Morning, students. So today we are going to study about this double fertilization triple fusion. As we are studying our chapter sexual reproduction in flowering plants, and in this we are going to study about the process of fertilization, syngamy, and then embryonic development in case of angiosperms. So in angiosperms, if you observe this double fertilization triple fusion, that is its characteristic feature. Characteristic feature means only angiosperms have this phenomena of double fertilization triple fusion. We cannot observe this phenomena in any other plant group. Even in gymnosperms also, there is no double fertilization triple fusion. It is only the characteristic of angiosperms. So now we come to the animation related to this. First, we will try to understand the process and then we will come to the application part. So if you observe any angiospermic plant body, here we find that this flower is the condensed shoot which is modified for sexual reproduction. And for that reproduction, it is having two important parts, the stamen and the carpel. If you see the carpel, the basal part of carpel is having ovary, and inside that ovary there is ovule, and inside that ovule we find the formation of the female gamete and all. So how this process is taking place, that we are going to study here. First of all, the cell that we are observing, it is the megaspore mother cell. Means now we are coming to the process of megasporogenesis. In this process, first of all, a normal diploid megaspore mother cell is there, and that megaspore mother cell is now undergoing meiosis. Due to meiosis of this megaspore mother cell, there is formation of 4 haploid cells. But out of these 4 haploid cells, normally 3 cells get degenerated and only one survives. So the cell which is now surviving is the megaspore proper. As it is the product of meiosis, so it is a haploid cell. So this haploid cell is there. It is having the nucleus with haploid set of chromosome. Just after formation of this megaspore, now it undergoes 3 repeated mitosis, and this mitosis is involving only karyokinesis. Means 3 repeated mitotic karyokinesis is taking place. So it will result into formation of 8 haploid nuclei. So here at this point now you can see there are the 8 haploid nuclei, and all these have been produced through the 3 mitoses taking place in the haploid megaspore. Now there is certain rearrangement, some morphogenetic movements is there, and it is working for rearrangement. Means the 2 nuclei come at the center and 3 nuclei move to opposite pole. What are the poles here? The one pole is of micropyle and another pole is of classa. So classal pole and micropylar pole, 2 poles are there, and they each having 3 nuclei, and at center there is 2 nuclei. Now there is formation of cytokinesis, formation of process of cytokinesis, that is resulting in formation of 3 cells at the classal end as antipodal cells. So these 3 white cells that you are observing, these are the 3 antipodal cells. At the opposite pole also, now you can find here the cytokinesis is taking place. It is also resulting in formation of 3 cells, but there is a difference in these 3 cells. The red color represented here is the egg cell, and 2 side cells, the lateral wall, these are the synergids. So these 3 cells taken together makes the egg apparatus. This egg apparatus is having 1 egg cell and 2 synergids. But there is no cytokinesis at the polar nuclei. So polar nuclei remain as it is. So this way, if you come to this entire structure, it is said to be a 7-celled 8 nucleated structure, that is the embryo sac. 7-celled, 8-nucleated embryo sac. Now the question is, where are the 7 cells? Here, 3 antipodal cells, 3 egg apparatus. So 3 plus 3, 6. Where is the 7th cell? That 7th cell is nothing but the embryo sac itself, because all this embryo sac, this formation has taken place in megaspore, and that megaspore is a cell. So that is the 7th cell represented here. But if you come to the nuclei, the 3 nuclei in antipodal cells, the 3 nuclei in the egg apparatus, and 2 nuclei as the polar nuclei at the center. So these are the total 8 nuclei. So 7-celled, 8-nucleated structure embryo sac is formed. This formation has taken place inside the ovule, in the ovary, at the base of carpel. Now we have to come to the male part. So related to this male part, we are aware that anther is the structure which is working for formation of pollen. So in that anther, through the process of microsporogenesis and all, there is formation of a large number of pollen. Each pollen is found to be a two-nucleated, two-celled structure. Normally, a pollen represents 2 cells. These 2 cells are the vegetative cell and generative cell. Larger vegetative cell, smaller generative cell. Through the process of this pollination, whatever means is there, whether it is carried by air or carried by water or carried by any animal, that pollen came to the stigma. So this transfer of pollen up to a stigma, that is called as pollination. So suppose this pollen landed on a stigma. Now the change is taking place. Now there is elongation of the intine, not exine. So now this intine is getting extended through the germ pore. And now this extended part is called as the pollen tube. You can find its extension through the style part of the carpel. In this structure, you can see that there is presence of 2 cells here. So when it is moving, one important change has taken place here. That change is the vegetative cell has been completely degenerated, and the generative cell has undergone mitosis. So it has resulted in formation of 2 male gametes. So now these 2 male gametes, through this pollen tube, now it is penetrating into the ovule. When this penetration is taking place, the most common method of penetration is porogamy. But in some cases, it may be clasogamy, mesogamy. So through any of this method, now this penetration is taking place. So suppose as this porogamy is most common. So in this diagram, we are representing here this penetration of pollen tube through micropyle. So through micropyle, it entered. Now you see after entry, what is going to happen? These 2 male gametes, now they are going to work independently. You can see on movement of this male gamete, now the synergid is getting dissolved. Here they also work as the filiform apparatus to provide passage for the gamete. Now one male gamete, see here, fused with the egg cell. That is the normal process of fertilization, synegamy. So here the synegamy is taking place, in which one male gamete is fusing with the female gamete, that is egg cell. But you know, one male gamete is still there. So what it is doing? Now the second male gamete, you observe here, it is moving towards the polar nuclei. And now it is fusing with the polar nuclei. Here in this process, you can find that the process is taking place at 2 levels. So it is the process of double fertilization. At the same time, it is triple fusion. Here these 2 polar nuclei and one male gamete, that is undergoing fusion. So this fusion product is primary endosperm nucleus. And as it is the product of fusion of 3 haploid nuclei, so definitely it will be triploid. And what about this? The male gamete and egg cell, definitely it is a zygote. So this zygote is diploid, primary endosperm nucleus is triploid. So these 2 different types of structures are formed here. During the process of embryonic development, finally the zygote will develop into the embryo, and this primary endosperm nucleus that will develop into the endosperm. So this way, this whole process has taken place. We have understood the process of this double fertilization and triple fusion. But now, understanding is one part. The second is here related to our questions. What type of questions can be asked? So we should come to the questions here. And if we come to these questions, let me share one question file here. So if you see a question here, I hope now this question is visible to you. So here in the first question, you can see that if a tetraploid female plant is crossed with a hexaploid male plant, then find out ploidy level of, first is embryo. Means here first we have to find out the ploidy level of embryo. Now come to the basic point. Embryo, how it is formed? Embryo is formed from zygote. How the zygote is formed? Zygote is formed through fusion of one male gamete and one female gamete. That is very simple. If you come to here, this female plant, that female plant is tetraploid. It clearly means that if this tetraploid will be undergoing meiosis, then it will form 2N. So the female cell will be 2N. But here, if you see the male part, it is hexaploid, means it is 6N. And if 6N will undergo meiosis, it will form 3N. So from this observation, what we can find, the male cell is here found to be triploid and the female cell is found to be diploid. So 3N plus 2N, it clearly means the zygote is going to be here 5N. So in this condition, the zygote is here 5N. If zygote is 5N, then definitely the embryo is also 5N. So we can easily find out that the embryo in this condition will be 5N. Now the second question is related to endosperm. So now we should come to the endosperm. How this endosperm is formed? We just studied it is through the triple fusion. So for this endosperm, how many male cells is involved? One male gamete is involved. How many female cells are involved? Two polar nuclei, and polar nuclei is part of embryo sac, means female cell. If you come to this one male cell, that is found to be here triploid, and what about this female? 2 into 2N. So overall, if you add here, what we can find, 3N plus 4N, so 7N is the ploidy level of endosperm. So in this case, the endosperm will be found to be 7N. Now the third part is also here. I am coming to that third part. The third part is here perisperm. So first of all, we should know what is this perisperm. Perisperm is nothing but persistent nucellus. In some plants, like the black pepper, there is persistent nucellus, and that structure is called as perisperm. So in this case, if you come to nucellus, now we should understand the ploidy level of nucellus. Where is nucellus found? Suppose if you are observing this ovule, so suppose it is the ovule here. Inside this ovule, suppose it is the embryo sac. In between these, in between the integument and this embryo sac, there is presence of some cells, nurse cells. They work for nourishment of developing embryo sac, and these are called as nucellus. So the ploidy level of nucellus will be same as the ploidy level of the female plant. In normal condition, it is diploid, but in this case, the female plant is already given 4N. So in the question it is given, that the female plant is here tetraploid. So we have to consider the normal ploidy level of the female plant, as the female plant is here 4N. So nucellus is also 4N, and if nucellus is 4N, then definitely the perisperm will also be 4N. So in this question, if you come to the final answer, the embryo, it will be 5N, endosperm 7N, perisperm 4N. In previous year AIPMT or NEET question, you can find these two parts, embryo and endosperm. But periosperm is not asked. So I have added one question here. In future, it may be asked. So we should know about the periosperm also along with embryo and endosperm. Based on this question, now you should do one home assignment. In this home assignment, what will happen in opposite case? Opposite case means what? You have to take the vice versa. In the question that we solved right now, in that case, female was 4n and male was 6n. Now, just you reverse it. Female 6n and male 4n. And you try to solve the same question. So that will be the part of your home assignment. You do it as homework. So that's all for today. And tomorrow we'll be studying something more about the same chapter. Thank you, students. Thank you very much.